My name is Justas, and I'll try to shortly introduce what we have done with data sets of protein and protein nucleic acid interactions. Uh, so as we all know, proteins interact with each other with short linear motifs of other proteins, which may be called peptides, and with nucleic acids. And these data are crucial to understand protein function and how they live, what they do. And to facilitate this analysis, we some time ago we developed the PPI3D resource. It's a database and a web server. So we take the data from the protein data bank. We do it weekly. We find all the interaction interfaces and we cluster them by sequences and structures, and then do user interface to use this data. So the clustering of the data is the crucial step in our pre-processing, because in PDB the data are usually very redundant. If there are almost 500,000 proteins, if you take the unique proteins, it's just around 100,000. And so basically, if some protein, if a structure of some protein is easily solved, people do it many times, and it's there many times. And it's usually done very simply. You just filter by sequence identity, and you get the non-redundant set of protein structures. But it's the different case with the interactions, because sometimes proteins interact in different ways. The same proteins interact in different ways. As you can see, this green protein, when it binds a ligand, it starts binding to a different site of the blue protein. And therefore, we clustered everything first according to sequence similarity, and then by structural similarity to get these alternative interactions. And it effectively reduces the redundancy. Here you see all protein-protein interfaces, about 1 million interfaces in PDB. But it's way less non-redundant interfaces, and these green ones are the alternative ones, which would get lost without this structure-based clustering. So we did all the clustering, we prepared all the data, and 10 years ago we first released it as a web server where you could introduce your sequence and or PDB ID and find the data on the interfaces and so on and so forth. But then we realized we are sitting on a huge pile of data which we cannot analyze ourselves. So we decided to make a simple user interface to retrieve everything. So you just add your own criteria, like the types of interactions you're interested in. We have not only protein-protein, but also protein-nucleic acid interactions. The sizes of the complexes and so on and so forth, and then you get a big CSV table. You can download the structures, you can download additional data, not very well documented, and then you can do everything whatever you want. I tried to group it into like three possible applications: large-scale analysis of data, new methods for protein-protein docking, and training machine learning. There are some examples. For example, in large-scale analysis, when I first presented this database, Susan from US came to me and said it would be interesting to analyze if there are helix dipole interactions in the interfaces, because alpha helices they have this dipole. One end is charged positively, other end is charged negatively, and they started analyzing it, and turns out in protein nucleic acid interfaces these helix dipoles are quite a frequent thing. Another thing, my colleague Clement, we work quite a lot on protein interaction modeling, structure prediction. Of course, there are AlphaFold scores, but we want to have some complementary scores that are not related to AlphaFold. So what he decided to do, he took all the heterodimers, not all, a non-redundant set of heterodimers from the PPI3D, and he redocked everything and trained a model on these good and bad docking models to predict whether the interaction interface is good or bad in the structure. It worked quite well in CASP. And the third application, we are still wanting to have some AlphaFold independent scoring for the structures. And we got back to the idea of statistical potentials. And we had in the lab VoroMK statistical potential based on contact areas. And we decided to train it on also for nucleic acids, ligands, and then make it all atom as it is popular now. And for the interaction interfaces, PPI3D clustering is used to analyze the data. And by the way, there is a poster on that. So that's it. There are some things that should be changed probably, because now the database is focused on binary interactions, but I don't know, probably the whole protein or protein nucleic acid complexes are interesting for people. Also, it is a very protein-centric resource. I think we should start moving into interactions involving nucleic acids. And if you have any questions, if you have any requests, you can ask me. And there is also a poster. So I have to acknowledge all the people who contributed. And that's it. Thank you very much.
Questions?
So far, do you have any reason to believe that antibodies or like subsets of antibody-containing complexes, like antibody-peptide complexes, behave in a way that's somehow atypical or not like conforming to the behavior of other types of complexes?
Yes, I would like to have antibody-antigen complexes as a separate category, but I didn't find a way to do it very fastly and conveniently. If you have some ideas on that, how I could mark antibodies, I get a structure from PDB. I have I don't know, several hundred of structures every week. I get a structure. I need to mark antibodies reliably. So if you are aware of any such method, if you could tell me, I would be grateful.
Thank you.
I think we may have time for another quick question.
I think the top, why don't you include the protein small molecule by the side?
That's one of the future works, probably.
Okay, thank you.